moving on to the Cahill cycle, or also known as the glucose-alanine cycle, or simply just the alanine cycle. It involves the amino acid alanine and the sugar glucose, where the muscle proteins are broken down into amino acids. These amino acids produce ammonium, which are toxic in high amounts, and to safely transport these ammonium ions to the liver, the body converts pyruvate into the alanine via the enzyme alanine aminotransferase, or ALT. Alanine is then carried by the bloodstream to the liver, and this cycle plays an important role when muscles are under intense stress, whether it's due to strenuous activity or lack of nutrients. The Cahill cycle takes place between three main sites: the muscle tissue, where glucose is broken down and alanine is formed; the bloodstream, which serves as the transport pathway for alanine; and the liver, where alanine is converted back into pyruvate for gluconeogenesis, and ammonium is removed through the urea cycle. The Cahill cycle serves several purposes, as it maintains energy balance during periods of fasting or long-term physical exertion. It supports amino acid metabolism and gluconeogenesis, the process of creating glucose from non-carbohydrate sources. It safely transports nitrogen from the muscles to the liver via alanine. It regenerates glucose in the liver, which is then delivered back to the muscles for energy. And lastly, it removes excess nitrogen from muscle tissue by converting it into urea, which is later excreted. So the glucose-alanine cycle shown in the diagram illustrates the exchange of metabolites between the muscles, bloodstreams, and liver. So in the muscle, the amino acids from the protein breakdown transfer their amino groups to glutamate, and through the action of alanine aminotransferase, or ALT, glutamate donates its amino group to pyruvate, forming alanine and alpha ketoglutarate. Alanine is released into the bloodstream and transported to the liver. So in the liver, the alanine is converted back to pyruvate and glutamate by ALT. The resulting glutamate releases ammonium, which enters the urea cycle for excretion. The pyruvate is used in gluconeogenesis, consuming ATP and NADH to form glucose, and this glucose re-enters the bloodstream and returns to the muscle, where it is used again in glycolysis, completing the cycle. The diagram also highlights the associated energy costs and redox changes in both the muscle and the liver. So to explain further, these are the processes with explanation on the chemical structures and how the bidirectional transfer of metabolites occur at the molecular level. So the processes in the muscles, the amino acid catabolism, the ammonia builds up during the breakdown of amino acids, especially during intense exercise, starvation, or muscle protein catabolism. So when the muscle breaks down proteins for energy, they produce ammonia as a byproduct. Ammonia is toxic, especially to the brain, so it needs to be removed from the body safely. But instead of releasing ammonia directly into the bloodstream, muscle converts it into alanine, which is much safer for transport to the liver, where the ammonia is removed and converted to urea for excretion in urine. The transamination reaction, or the alanine formation, what is transamination? The transamination in general is the transfer of the biochemical process where the amino group of amino acid is transferred to a keto acid. So, resulting in the formation of a new amino acid and a new keto acid with the transamination. So, sa muscle cells, ang glutamate mo-donate siya og amino group sa pyruvate and mo-form siya og alanine and alpha ketoglutarate. So, to start the pyruvate, naa siya carbonyl group and carboxyl group on carbon 1. So, ang glutamate, mo-donate siya sa amino group from the alpha carbon to the carbonyl group, of the pyruvate. So this replaces the oxygen of pyruvate's carbon 2 with an amino group forming alanine. So in summary, one nitrogen atom is transferred from glutamate to the pyruvate, which creates the desired product, which is the alanine, a 3-carbon molecule. The byproduct is from glutamate becoming alpha ketoglutarate by losing its amino group and reforming alpha keto group. The enzyme involved in this process or in this reaction is the alanine aminotransferase or the ALT. The substrates or the glutamate and pyruvate, these are from the glycolysis, and then the products is the alanine and the alpha ketoglutarate. So in the bloodstream, while there are no enzymatic reactions occurring in the bloodstream, it serves as a vital transport route between the muscle and the liver. So essentially acting as the bridge of the cycle. Now, the alanine transport, this is where the alanine is released from the skeletal muscle cells into the bloodstream. It travels through the blood to reach the hepatocytes, the liver cells, and this allows nitrogen to be transported safely without releasing free ammonia. So moving on in the process that is occurring in the liver, once the alanine reaches the liver, it is used to regenerate glucose and safely eliminate nitrogen through the urea synthesis. The reverse transamination, this is where the alanine is deaminated, releasing pyruvate for glucose synthesis and transferring its amino group to alpha ketoglutarate. This is the reverse of the transamination in the muscle. So in this case, the alanine donates its amino group to ketoglutarate and alanine becomes pyruvate and alpha ketoglutarate becomes glutamate. So just the same as the process earlier, the enzymes involved is still alanine aminotransferase, the substrates or alanine and alpha ketoglutarate, and the products are the pyruvate and glutamate. So the oxidative deamination, this is where the glutamate produced is deaminated, releasing ammonia, which is toxic. Ammonia is toxic, and it must be disposed. So first, the nitrogen atom on the alpha carbon is removed as ammonium, and then the alpha carbon reforms a keto group, regenerating alpha ketoglutarate. So the enzyme involved in this process is the glutamate dehydrogenase or GDH. The substrate is glutamate and the products are ammonium and alpha ketoglutarate. So the urea formation, this is where the ammonium enters the urea cycle. There are two nitrogen atoms from two glutamate molecules or other sources. It's combined with a carbon from CO2 to form urea. So the urea has two nitrogen groups and a central carbonyl group. The nitrogen excretion, the nitrogen atoms originally from muscle amino acids are now safely excreted via the kidneys in the urea. Now, in the gluconeogenesis, the pyruvate is used to synthesize glucose via the gluconeogenesis pathway. It is explained by Lanz to recover glucose. So, in the bloodstream, the glucose transport liver to muscle. After gluconeogenesis in the liver, the newly formed glucose is released into the bloodstream. This glucose is then taken up by muscle cells for energy through glycolysis. So the key enzymes and molecules involved are the alanine aminotransferase or ALT. It catalyzes the conversion between the pyruvate and alanine. The glutamate and alpha ketoglutarate, it acts as nitrogen carriers and participate in transamination. The urea cycle enzymes, it converts toxic ammonium to urea. And the gluconeogenesis enzymes, it regenerates glucose from the pyruvate in the liver. So the Cahill cycle is extremely important because it prevents toxic accumulation of ammonia in the muscle tissues. It supplies muscles with energy during times when oxygen or glucose is limited. It supports the liver's role in homeostasis, especially through gluconeogenesis and the urea cycle. And it also plays a central role in protein catabolism and the nitrogen disposal. So, without this cycle, the prolonged exercise or starvation would lead to dangerous ammonia levels and muscle fatigue. So, that's it for my topic. Thank you very much for listening.